All right. Thanks everybody for joining. Welcome to the ATP-Bio Public Webinar Series edition for June. Super exciting topic, which is startup companies, and we have a great panel to join us. For those who haven't met me, my name is Dan Chen. I just took over from Tim Tripp, who as the interim strategic partnership and innovation director based here in at the U of M. So. It's a pleasure to be part of this group. I'm on day 5 of the job, so if there's glitches here or there, make mistakes, don't know what acronyms are, please bear with me. So we have a little bit of housekeeping to do before we get started on the actual panel itself. Going on that. So this is one of a series of public webinars. These are open to anyone. And again, thanks to the Society for Cryobiology for promoting this event as well. I have some information about what they're doing too, in terms of their webinar series. And certainly the annual event coming up in DC in July. So again, we've got a link there to register for that. Should be a really exciting conference, and certainly a great opportunity to meet others in the cryobiology sphere. So for those who are joining as part of ATP-Bio itself, a couple of things to be aware of. We're in the middle of the year 4, and we're doing a progress update. That'll happen this week. Alyssa Berger will send invitations out for that. There's also the DCI Cafe, which is running on a regular basis, and we also have then the closed forums. We'll be talking about kind of the progress on the convergent research, kind of connection to the other pillars, and so forth. So stay tuned for that. So it's an exciting year, obviously, for ATP-Bio, going through as a renewal year this year. So with that out of the way, just wanted to introduce our 3 speakers today. This is just a great panel that's been set up. I have to thank Tim for putting this together. We have 3 speakers. I'll do quick introductions of each of them, and what we'll do then is have each of them talk a bit about themselves, about their company, anything else they want to share. If you have questions, I'd have you put those into the chat, and then we'll moderate those at the end of Kevin's presentation. So hopefully that works for everybody, and we'll have kind of like expect 10 minutes or so for each of the speakers to talk about themselves and their companies. Then we'll have the remaining time just as a panel, and I'll moderate the discussion. But if you have in the chat, if you want to direct it towards a specific person on the panel, that'd be great. Otherwise, we can open it up to all 3 speakers. I'll say just a couple comments about each of these speakers, just to introduce them. These are really well-established, highly accomplished individuals. I won't capture everything they've done and use a lot of our time. I'll start with Matt. Matt Powell-Palm is the CEO of BioChoric. He's also a professor of mechanical biological materials engineering at Texas A&M. Did his studies at Carnegie Mellon and Ph.D. at UC Berkeley, I think with Boris Rubinsky, if I got that correct, Matt.
Yep.
Mehmet Toner, you should all know, is our deputy director for ATP-Bio. Also professor of biomedical engineering at Mass General Hospital, is part of Harvard Medical School, member of National Academy of Engineering, which certainly speaks for a lot of his academic background. But I think many of you are not familiar with the fact that Mehmet has also started several, co-founded or founded several startup companies, very successful, and certainly brings both that academic and commercial experience to bear. So, and last we have Kevin Flynn, who is the Chief Science Officer for CaseBioscience. Kevin did his training at Loyola and Colorado State University, and a postdoc we were talking about earlier at the Max Planck Institute. Kevin also has some broad industry experience, having spent many years at Biotechne, so can certainly bring experiences from both large company and small company to bear on the discussion. So with that, I will stop sharing and I will turn it over then to Matt. And Matt, I don't know if you have slides to share or you just want to just have a discussion, up to you on how you want to talk about yourself and your company. So.
Yeah, I've got just a couple of slides that I threw together as a little bit of a visual guide. I resent that you stole my introduction in bio, Dan. Geez.
I was going to ask you about the hammer incident that was quoted in the latest UC Berkeley website about. But anyway, I'll let you get started on that.
Yeah, sure. We can talk about the hammer. Let's see. What are you guys seeing? Are you seeing this PowerPoint? And is it
we're seeing a presentation, but not there? It is now. It's
okay.
Now it's good. Perfect.
Okay, so yeah, hi guys. Looking at the people on the call, I think I know many, if not most of you. So I'll try and keep this introduction brief. I also think, I by far and away have the least to offer to this conversation as compared to Mehmet and Kevin. So very much looking forward to hearing what they have to say and getting right to it. So just as a brief overview to complement Dan's already excellent bio, I kind of wear, or I should say, in my capacity as a speaker here, I'm wearing sort of three different hats. As Dan mentioned, I'm a professor at Texas A&M University, and then I'm the co-founder of a biotech startup called BioChoric. And one other thing I'll mention too is that this is a this webinar is co-hosted by the Society for Cryobiology, and I'm also on the board of governors for the Society for Cryobiology. So I try to keep my fingers in a number of the relevant biopreservation related pies, if you will, on both the sort of academic side and the service side and the industry side, which I think has proven to be a useful strategy over the years, because we're working in a field right now that is so young relative to its industrial translation, right? There are all of these exciting new, potentially translatable biopreservation parallels that are coming out of academia and government and institutions like the Society for Cryobiology. So I'm constantly trying to maintain sort of the dialogue between all of these different potential policy, industry, and scientific actors in the name of making sure that what we're translating is at the absolute cutting edge of what's out there. So let me just give you a sense, especially for maybe the younger folks, the trainees on the call, of what my own timeline was here and is here. I did my PhD with Professor Rybinski at Berkeley from 2017 to 2020, which is where we sort of invented most of the platform technologies that now power my company. And something I'll note, I don't know if there's any other big academic family tree nerds in the audience, but Professor Rybinski's lab has produced a number of really interesting actors in the biopreservation space, whom you all know. John Bischoff, one of the early students of Boris Rybinski, and Charles Lee, and John Ed over there at MGH, and then Boris came from the same lab that Mehmet Toner came from. So in the sort of physical science thermo side of biopreservation, it's actually kind of a small world and a tight-knit family. That always intrigues me, so I pointed out. Okay, so then after I finished my PhD, I started a postdoc, and during this postdoc, I founded this company, BioChoric, which I'll talk a little bit more about. At a high level, the purpose of the company was to begin the process of commercializing the sort of scientific advancements that were the product of my PhD. So this is a part of a philosophy that I inherited from my advisor, which is that, it's so easy as an academic to spend your entire life writing excellent papers that describe findings that then never make it out of the lab. And so the question is often, how do we make it so this stuff that we've put so much time and intellectual energy into, how do we get that out there into the world so that it can start helping people and benefiting society? And so Boris's attitude and now that which I've adopted is that you kind of got to do it yourself. In some select cases, you'll get really lucky, and 3M or somebody will come by and want to license your technology, and then they can handle the translation. But more often than not, the people who have to get these sort of new, fresh ideas out there into the translated industrial space are the people who invented them, right? So that was sort of my attitude in founding the company. And then after my postdoc, just 2022, 2 years ago, I scored a tenure track position at Texas A&M, which is a top flight R1 research institution. And I want to note that, my ability to land that spot in the unbelievably competitive tenure track professor market right now was due in large part to ATP-Bio. And so not only the work that I did under the guise of ATP-Bio, but also the connections that I built and the strength of the intellectual community that ATP-Bio has produced. So big shout out and thank you. And you know, to any PhD students or trainees or postdocs who are looking at faculty positions right now, I mean, you know, be confident that the work you've done at ATP-Bio will be extremely helpful in helping you to find a job in the academic space where you can keep pursuing this sort of fundamental scientific work. So I'll talk just very briefly about what BioChoric is. As are many sort of early stage scientific startups, our purpose is varied and nebulous. But generally, what we're working on is developing the technological foundations for a new global cold chain that is based on ice-free biopreservation technologies. So the sort of core IP portfolio that we're commercializing involves 15-ish patents in the isochoric biopreservation space. So using biopreservation techniques in various thermodynamic regimes that occur under rigidly confined conditions. So we build isochoric chambers and then various thermodynamic protocols that occur in those chambers to accomplish various, let's call it biopreservation goals, including, you know, preservation of whole organs for transplant, preservation and treatment and sterilization of various fresh food products. We're working a lot these days in the cryoconservation space, you name it. But anywhere there's intersections of the finite shelf life of life and industry, we're trying to get a foothold and apply the principles of isochoric biopreservation. So a few salient details about the company that may be of interest. So it's a UC Berkeley spin out. So all of our, or not all, but a good portion of our IP is anchored through the UC Berkeley Tech Transfer Office. And I really can't recommend enough for small startups to build a strong relationship with your university tech transfer office, because it gives you connections and legal infrastructure and all kinds of exciting capabilities that otherwise you got to pay a lot for, you know. We, over the course of the last, I guess, 14 months, have scaled from one full-time employee to about 10 full-time employees. So we're in that sort of exciting growth period where we're now starting to do a lot of in-house work. We have locations in Bozeman, Montana, and Berkeley, California, and down in Houston, Texas. We sponsor a bunch of postdocs, both at UCB and then in a couple of different government labs. And we're in various stages of translation in a number of different sort of exciting biopreservation spaces that can loosely be categorized as biomedical in nature or agricultural in nature. And so just to give you, you know, a fun little snapshot of a couple of these things, we work a lot on transportable, cryoprotectant-free supercooling of transplantable organs. So we're in the middle of a large preclinical push here with the Texas Heart Institute, doing full orthotopic heart transplant trials, powering some initial FDA conversations. We work largely through private conservation funders on the cryoconservation of a variety of endangered marine organisms. So we started working on coral with Smithsonian National Zoo, and now we've expanded into working on endangered in amphibian models like the Wyoming toad and various endangered mammal models. And then finally, we do a ton of work. I'd say about half our scientific staff is dedicated to interesting applications of low temperature, high pressure thermodynamic protocols that are achievable in isochoric chambers to the reduction of fresh food waste. So this is, you know, I think a critical societal problem that a lot of people in biopreservation maybe don't think of as one of the first applications of our general suite of technologies. But about 40% of the fresh post-harvest food generated in the world ends up going to waste, even while global hunger is ballooning and the amount of water and carbon that went into making this food was extreme. And so, if you're interested, if you're a biopreservation person and you're interested in the sort of climate tech or global health play, there's the obvious medical applications, but then another really exciting subdomain where our intellectual community can have a big impact is in improving food cold storage to reduce food waste, especially of really sensitive, high water content fresh food products. Think tomatoes, spinach, sensitive fruits and vegetables. These are things that very often wind up mostly in the trash, whereas with just slightly superior technology and technologies that leverage a lot of the same principles that are relevant to medicine, we could be reducing a lot of this food waste. So this gives you a little snapshot of a few of the scientific domains that we mess around in. And with that, I'm really happy to dialogue and answer any questions after we hear from the other people. But in the meantime, let's move on to Mehmet and Kevin and hear the good stuff. So thanks very much Dan for having me.
Thanks, Matt. Appreciate it. Really exciting. I'm sure there's going to be a lot of questions coming in the chat, but about your experience with BioChoric, but great to see that. Mehmet, I'll turn it over to you, and you can go ahead and talk about whatever you like to talk about in terms of your startup experience. So,
thank you, Dan, and thanks for the presentation, Matt. It's wonderful. And just so everybody knows, Matt mentioned Boris Rubinsky, and we all come from almost the same family, and the giant of that family is here actually, and Ernie Kramalho's first graduate student who invented the modern cryo microscope, a good friend, colleague, and mentor, Ken Diller. And so this all started with him. If you guys don't like it, just blame him. And I don't have any slides. I just, and I actually want to be short so we can have questions. And just very briefly, and I won't just stick to cryobiology, my journey about commercialization and startup. I've never been in a startup myself, so I'm not an expert. I just started a bunch of companies in my long academic life. And I feel cryobiology is in a very opportune time, and Matt really laid it out so nicely in the sense that there are many very important unmet needs, both clinical and global health, as well as food, agriculture, biodiversity, that really rely heavily upon solving problems of supply chain of living products. So you can't supply living products unless you ship them properly and store them properly. It's not going to happen. Otherwise, we're going to have cell therapy for $500,000 per dose of therapy. And my journey started, I wasn't interested in startup like Matt the day I started my academic life. I was told to do otherwise. And so my journey in commercialization started in early 2000s. And I'll give you examples of two companies. The first one we started, we are engineers, so we always come up with the most uninteresting name. And it was called Living Microsystems. Because I was doing microsystems and microfluidics, and I thought that would be a good name. And that company was interested in prenatal diagnosis. And in about 20 years later, it changed the name to Verinata Health, and it was acquired by Illumina, which is a very big current clinical genomic company, one of the largest, probably the largest genomics company. And they acquired it in 2013, and it's used in 70% of all pregnant women today. And so that's one reason why it's fun to do translational work, because if you succeed, the satisfaction is tremendous. They wouldn't even know my name at Illumina, but they are using my patent. And but that's one, that was the first one, the successful one, and it's been very difficult since then. And the perils of commercialization is amazing. It comes from right and left when you don't expect. It's very important to structure the companies properly, and it's very important to make sure the risks are eliminated as you move forward and you build value. Another one that we started, that's one that I'm very proud of, but it failed. It was for global health, for HIV/AIDS diagnosis, called Daktari Diagnostics. Daktari means doctor in Swahili language. And the company was very successful. It was doing like a blood glucose test. You take a finger prick of blood, you can measure CD4 cells. It was the first point of care test that was ever developed that will do a counting of a cell at a point of care, disposable $5 device. And it worked as good as a modern flow cytometry, and it worked. It's been tested in 4 countries in Africa with 3,000 people in flying colors. Absolutely no issues. Technology is working like we have better than we imagined. And $40-some million raise for the company. Everything was looking good until one morning we woke up and read a little white paper from World Health Organization that said that we don't want CD4 test anymore, we want the viral test. It was not a good idea for World Health Organization to do that, because they still don't have viral tests, but the company bankrupted, went under, because the investors wouldn't continue with that kind of a major risk. So here we go. You have a working technology, but the company didn't do well. We have a number of companies in cryobiology. Silvatica is one of them for organ preservation, and few others. But I'll answer those questions, hopefully, when people ask questions. The one thing, especially for the young people who are trying to decide and whether they want to start a company, if you do not have entrepreneurial gene, do not start a company. And it is a long road. It's the most exciting. I think it's more exciting in academia, if you ask me, actually. But be ready to fail. A failure is not an option. It's a requirement. And it's just that if you know how to fail, you succeed in the end. You pivot to better and bigger things. The journey and the team are the most important things. And the other advice I have, please don't be in love with your engineering. Once you start a company, you make sure you have an IP you protect, and make sure there is a market. Make sure you structure the company properly. And it is, many of us are trained in doing things so well, and in engineering, you think building the best device is going to make a company successful. I just gave you an example. We built the best device, $47 million, and it did not succeed. So with those notes, I would say just, it's the most exciting thing is to see young people forming companies and succeeding, and it's, happy to answer questions.
Great, thanks, Mehmet. That's a great experience, and interesting, especially sobering too, to see how technology doesn't always win. But I'm sure we'll get to questions about that and your experience in a bit. With that, I will turn it over then to Kevin. I know, Kevin, you have a couple of slides to share, and you can talk about your background and what you guys are doing at Case. So thanks.
Sure. Can everyone hear me okay? Yeah, so
Yeah,
thanks for the invite. I'm not going to talk too much about myself because I'm probably the least accomplished panelist here today, but they needed to fill that third spot with somebody, I guess. So here I am. So here I do have some slides. I want to introduce you to the company. So I'm probably on the extreme side does everyone see my screen okay? Okay, yeah, so I have a bunch of slides. I'm going to move quickly, but I just want to give a little bit of a different perspective, more from kind of a hybrid approach, from looking at it from the business perspective of starting a company, as well as the science and regulatory. So these are all considerations that I think went into the decision to start CaseBioscience. So we are CaseBioscience, and we focus on biosolutions, so media and biopreservation solutions. So we were started with the vision to have a quality-driven ISO 13485 facility to formulate advanced media and biopreservation solutions for the advanced fields of cell therapy, regenerative medicine, and human ART. And standing for Assisted Reproductive Technologies. So a little bit about our approach. So we take key learnings from art. So our founders, and our foundational scientific team, our foundational team, all come from art. So from the assisted reproductive technology field. And this has a rich history of undergoing a lot of scientific changes and regulatory changes over the years, especially in like the 90s and early 2000s. And so what we're doing is we're really taking key learnings from art and applying it to cell and gene therapy. Particularly on the regulatory experience and also on the cryopreservation side, like non-DMSO cryopreservatives have a deeper history there. And also on the other side, on the flip side, we're also taking key learnings from cell and gene therapy. There's a lot of new research in regarding with pluripotent stem cell culture, and we're applying those ideas back to looking at maybe improving some of the more established protocols and solutions in art. So it's a really reciprocal knowledge transfer. So why start a company in biotech, and why particularly art, and why CGT? And this is something that is maybe obvious to everyone, but it's something you got to consider, and that's this market analysis. So you can look at any of these projections of the art market and cell and gene therapy market and just see that they're growing at a clip. So assisted reproductive technologies is already a big market, and it's growing probably at a rate of about 9 to 10% per year. But the cell and gene therapy market is a little bit smaller now, but is really growing at this 20% CAGR. So there's a huge market. And so how could CASE contribute? So our founders went and looked at this, and they had a lot of history in the art field and looking at cryopreservation. And really what is a limiting factor is for the widespread adoption of cell therapies is the limited options and suboptimal cryopreservation for some cell types. So in a lot of cases, current DMSO solutions are not sufficient. There's really a need for efficient, cost-effective cryo for the widespread adoption of cell therapies, especially if you think about allogeneic therapies, right? There's one dominant solution out there. It works, but it may not be the best for everything. Issues with DMSO include toxicity, cell dysfunction. So really there was a need for new cryopreservation solutions. And many of the people in the audience are working on this as well. And cryopreservation, the market is growing, and it is important in many different areas, as Matt and Mehmet pointed out. But it's a part of, so this projection here is a big part of how cell gene therapy will succeed and how assisted reproductive technologies has succeeded in the past, and how many thousands of babies have been born has been based on cryopreservation. So just a little bit about why CaseBioscience felt like they could contribute something. As I mentioned, our management team came from two previous startups that were very successful. I just mentioned Monica Mazzezzi founded Life Global in the 1990s, and they really, as she started from a startup all the way to growing the company to a powerhouse in art. And they really focused on the one-step media, and I was talking to Mehmet about it. It was pioneered by Biggers back in the day, but then modified and really took to the long game. So you have to persevere in this field, I think. And after 20 years, they really became the dominant media for growing human embryos for assisted reproduction in humans. So this company that and as well as Evolve Gene, founded by our co-founder Ami, were sold in 2018. So then we also, in running that company, we have the experience of an ISO 13485 facility. Want to talk about that a little bit briefly in the future. We have a strong scientific team, which of course is very important, a global network of distributors and sales, and the regulatory experience. So I'm not going to go through this in detail, but this is just a quick timeline. We were founded in 2021. One important aspect was establishing, from my perspective, an R&D lab. And so we established that in late 2022, started with ideas that we brought from art into CGT. And this was very important, is acquiring this ISO 13485 certification that gives us the credentials to make medical grade equipment and devices. So with that, we focused on building a strong scientific team. We have a great board with a lot of experience in ART as well as in CGT. And so our company has these three kind of prongs, or a tri approach. So we have the cell gene therapy market with a focus on cryopreservation, assisted reproductive technology, and contract manufacturing services. So I'll talk a little bit more about how we also looked at the market a little bit, looked at the competition to successful companies, and saw what their portfolio was offering. And we offer a similar portfolio, and but really wanted to improve upon some of these things that were available. So when we go through a product and think about how to bring it to market, there's basically this basic workflow. You have your R&D, you have widespread sampling. So this is a period where you're not making any money, you're not concerned about that. You want to show people, get it in people's hands, and really validate it in other people's hands. So this is kind of the phase we're at now, before launching our products. And this is relevant to any of the products that we're considering. And you can see collaborations on the top. We are all about collaborating. And the cool thing, I think, is that cryobiologists are very collaborative, as well as our potential customers. And this RUO, so we're focusing on first research use only, and then looking at the clinical phase of product development. One thing I have to mention here is that the foundation of our company comes from this art background, where our products are classified as class 2 medical devices. So this is important because currently cell and gene therapy have no official designation, but there is FDA guidance, and we kind of foresee, or our founders foresaw, that this was going to be important in cell and gene therapy. So we focused on the quality management system, getting ISO 13485, which has a risk management plan, a design and development plan, as well as a manufacturing plan, all under these criterias of making a medical device. And so this is where I spend a lot of my time, is in this area here, since I run the R&D. It's a bit onerous, but it's essential to have the trail, the paper trail of everything you do. And that's really captured in this design history file, which is required by the FDA for medical devices. So it includes all steps of the process. So from that, we have this platform that's based on this reciprocal knowledge transfer between ART and CGT. We have something we call CaseBase, and we use that as the platform to derive other products. And one thing that we strive for is high quality, of course, conforming to the guidelines for cell and gene therapy, as well as looking at consistency and accessibility. We focused on making things easy, like passive cooling techniques, and then, of course, performance. And as everyone in this audience knows, cryopreservation is extremely important for cell therapies. I'm just pointing out, of course, that the different steps, whether it's a cell isolation, or in the case of pluripotent stem cells, the pivotal cell type is the pluripotent stem cell, from which you derive the other cell types that are important for the various cell therapies in regenerative medicine. So we focused on pluripotent stem cells first, and again, the link with ART is that it is the inner cell mass of the blastocyst. So that is another point to make here. And pluripotent stem cells, of course, give rise to all the cell types. We have developed a non-DMSO, which is working extremely well, and then also a DMSO solution. I'm not going to talk too much about that. We're data-driven to show that these formulations work. We also look at the whole workflow and having a synergistic workflow. And then, so just on the flip side, on the ART side, there's also cryopreservation is extremely important for the gametes as well as for the embryo itself. There are commercially available solutions that work, but there are improvements that can be made, and we won't go into the details there, but I think some of the people in this audience know as well as I do, if not better. And we have a rich history. So James Stachecki has developed these formulations over decades and have a lot of clinical data, but we're making improvements, making them protein-free, for example, and things of this nature. So I just want to mention some key learnings after about a year of our business being operational. The idea of collaboration has come up time and time again. I think we all listen and learn from our scientific advisory boards, and even collaborating with other cryobiologists. Even though every cryobiologist I think I've met is super entrepreneurial in their own rights, they're willing to collaborate and find room for synergies between different businesses. And also listening and learning from your customers. So we've gone to like facilitate and a lot of these cell and gene therapy conferences, and learning about what they want, the costs, the cell-specific survival, things like that is very important. Also, there's a hunger for novel developments in cryobiology. I think everyone in ATP-Bio knows this, and it's good to have that reaffirmed by people who are not cryobiologists but need it in their workflows. And an interesting thing that I've learned is that cell and gene therapy scientists in particular are sophisticated in cryo. They're willing to try new techniques. They're willing to use control rate freezers and try really nuanced protocols just to get things to work well. And also, there's a need for innovations in art. So a lot of people consider art a established field, but really there are rooms for improvements. And we also have listened and responded. We've changed our portfolio over the years to focus on certain things like the ART products. And so this is a little bit out of cryo, but looking again at the embryo culture. And we've made some improvements there, and we're really excited to get to a protein-free blastocyst growth media, and with improved blastocyst quality, which is really substantial when you think about that these are potential human lives. So that's kind of us in a nutshell. We're all about collaborating, and yeah, that's it from my side. So thanks for listening to my spiel. I'm gonna stop sharing.
Great, thanks, Kevin. Appreciate the background and then the detail about Case. Very, very exciting. With that, we're going to go ahead and open it up questions and start with kind of some questions that have come up in the chat. Do you want to do a special shout out to the team out at UC Riverside? I think Catherine, you're with that group there. Bunch of students who are interested in academics as well as startups and so forth. So glad to have you guys join. So with that, I'll just start picking up a couple from the chat and then some other ones that are maybe more general. First one is very specific for you, Mehmet. It's from Don Rieger. A number of years ago, you with John Biggers showed successful fertilization with dried mouse sperm. Did you ever commercialize that?
Funny. And at the beginning of Kevin was asking about John Biggers, and yes, we did work on it. We never commercialized it. It worked actually very well, but desiccation of mammalian cells has been a tremendous failure, certainly in my lab, but in other labs too. No one can actually take a mammalian cell and store it at room temperature like a desiccated suspended animation in an organism. In this case of sperm, that's a little bit different because you inject the DNA into the egg, so the bar is much lower. So in that case, it worked. Yanagamichi is another person who really pioneered that area. We did it in a little bit different way. We never looked into commercialization.
Yeah, very good. Thanks, Mehmet. Good point there. We have one other one, which is kind of a question and a comment from David Orta about opportunities that community college students could take part in either your organizations or kind of the general cryo field. So I would just say if you do have those opportunities, speakers or anyone actually on this taking part in the webinar, just go ahead and chat that out. I think you chat it directly to David, or you can put it in the general chat. So thanks for that. I'll start off with a couple questions from my side, and one is, especially with a lot of people who are part of ATP-Bio who are coming from an academic setting, and this maybe start with you, Matt, on this question. How is the kind of the mental shift going from an academic type of mindset to a startup mindset? Is that something that has been difficult, or is it only, I think Mehmet talked about the entrepreneurial gene. How has that been for you, and have you seen that, translated for other people, and maybe some stories about that piece?
Yeah, In my experience, I think the entrepreneurial and the academic mindset are very, 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 very similar. And that, like, you know, the things that I have to do to keep my company afloat are in many ways qualitatively similar to what you have to do when you're a young tenure track faculty trying to keep a lab afloat. You know, you got to go find the money, manage the budget, and do the science all at the same time. You know, so the sort of the rat race element of entrepreneurialism is something that I think also exists in academia if you're going to go the tenure track route. Now, I will say, if you're not going the tenure track route, you know, if you're going to be you know, let's say you're coming out of a postdoc where you've been on that blissful couple of years where you get to do science unencumbered by bureaucracy or funding or teaching, then it's an incredible transition. Where, you know, I think the hardest part for me, which applies both to, you know, my business experience and my professorship, is how little time I get to spend, you know, staring out the window thinking about thermo relative to how much time I would like to spend. So, you know, I really appreciated Kevin's presentation, sorry, because it showed a lot of the, you know, the sort of hard realities of what building a business, especially in the life sciences, consists of. You know, there is a lot of regulatory formality and I don't want to call it bureaucracy, isn't the right word, but there's a lot of structural relationships that need to be built and maintained that are complementary to, but ultimately tangential to the science, right? So if you're coming at it from hey, I'm here because I love the science, it's a concession that you have to make. I don't know if that answers your question.
No, it does, and that's maybe a quick follow because you know you're doing both, right? So you're the big R1 university, you're on a tenure track position, and you've got this startup. Maybe how you think about managing, you know, both those priorities, or is just you're just working all the time? It's essentially, or how does it work out for you?
Yeah, I mean, an unfortunate reality of it is that I'm working all the time right now. But I'll also comment that, you know, our company is in the blissful pre-significant revenue R&D phases, where again, the sort of day-to-day realities of your academic science and your industrial science look very similar. You know, you're in the lab, you're doing experiments, you're writing papers, you're talking with collaborators, you know, you're doing all of these things to build out an intellectual platform that will support either, you know, your academic career or your company. And these things look more and more similar. Now, if you ask me this question again in you know 1 year or 2 years or 3 years, I think the tenor will change significantly. But in this early stage, it's all Gucci.
It's all good stuff. I like that. I'll turn over maybe to Kevin because you made a different transition going from, I think, you know, certainly from postdoc to then I think last at Biotechnique, going from big company to small company. How has that transition been for you and kind of, again, the mentality shift of being part of a startup?
Sure. Yeah, so it it was a shift, but it was a good shift. And like a question like, would I do it again? And yes, I would. So I worked at a big biotech company before. I was happy there. Worked with great people. Uh, but there's a little bit more bureaucracy at a big company and less ability to really have a seat at the table in terms of the direction of the company and the overall vision. So when I was presented with that opportunity and met Monica and Ami, the co-founders, and Don, the scientific advisor, I was sold on the idea of having a seat in the table and helping determine the direction of the company. The subject matter was also very important. Cell and gene therapy and assisted reproductive technology, and the idea of merging the two was very appealing. And seeing the experience of the team, it wasn't an easy decision, but I'm glad I made it. And you know, it's hard work, like Matt said. I mean, even though I'm not, I don't have tenure track physician. I'm an affiliate faculty at Colorado, but I have the luxury, I should say, of focusing on the business side and CaseBioscience. But it is very similar. I think, like Matt said, like the looking at the science, doing the experiments, but then also worrying, where's the revenue going to come and things like that. But yeah, and it's a little bit more intense than working at a big biotech company, but in some way, but more exciting at the same time. And being able to be part of something, you feel like you're more an integral part of what you're doing at a small company. So I hope that answers.
But I think what you do matters, right? It's pretty visible to everybody.
Yeah, it's visible to everybody, and everyone has teeth in the game. It just feels like there's again more collaborative spirit, more everyone is involved and invested in what you're doing. So
good, thanks. I'll turn maybe topic over to maybe a little bit different thematic, which is about startups in cryobiology in particular. And Mehmet, you've got experience in startups inside and outside of cryobiology. Any specific things you would say that make it more or less challenging to do a startup in cryobiology for all the people here who are involved in ATP-Bio?
Yeah, I mean, the question is for any startup company is how are you going to make money? What's the market? And a lot of what cryobiology type companies have, it's not the case for Kevin's company because that's a solution and it's a good market. But selling a chip or something to do something, and that doesn't really make a lot of money. And even if you capture the entire whole, let's say, cryopreservation of oocytes, if you capture the entire market for every single freezing device, it's still a tiny little market. And so raising money against those kind of ideas are very difficult. It's very important to find the right applications and market. And the good news about cryobiology with cell therapy, the areas that came, by the way, Kevin, you were not the last one they asked. I was the last one. You and Matt, the first two, and then they needed to, and because I'm the deputy director, they said, okay, Mehmet, you have to fill in the last
I
spot.
have no experience. I just was given this
Yeah,
agenda and these speakers.
yeah,
I have all three of you.
you came into this in the last minute, but so that's the market is very important. A lot of our postdocs and people who start these companies in the early phases are too much giving emphasis to technology. I'm not trying that technology is not important, but if your market and the structure of your company is not good, at some point you need somebody to put their hand in their pocket and give you a lot of money to make that product successful. And typically that's going to be anywhere between $50 to $500 million. And when people give that much money, they say, okay, what's in it for me? They are investing not to make you successful or happy, but to sell a product that's going to give them return on investment. And sometimes we forget about that. And it's very important. The structure of the company also is very important. A lot of the things we do in cryobiology are platform technologies. And like the cryotop or this or that, they could find applications in different areas. I will be very careful in structuring the company in a way that you don't lose nobody is able to go after every single application of an idea. And platform companies were very trendy 30 years ago, not anymore. I will structure in a way that is actually only for the application in the field of use and eliminate and exclude every single other application because they give you $1, they're going to own every single application unless you structure the company properly from the beginning. Do not touch professional money until you have the right structure. You will get burned badly.
That's really a really good piece of advice, because I think if I think about the field, I'm brand new to this whole field of cryobiology. There's so, and I think you had the chart, man, that showed all these different application areas, right? And I think that how maybe you can comment also on how you think about what the prioritization of those and how do you do assessments? How do you compare biodiversity to what's happening in Oregon? Because there's so many opportunities when you have a technology platform, right? And I think Mehmet's point being, this platform that can do a lot of different things. How do you think about that from a company and a revenue perspective?
Yeah, and I totally agree with everything that Mehmet said. And I think, there's two aspects. One, when you're dealing with a platform technology, as many of us are, something that we develop from often a physical science perspective and then find to be applicable to a wide variety of biological products. What you're sort of posed with is a solution in need of a problem, right? So a lot of the early machinations of a startup in this space are lasering down into the different markets and finding exactly which of these applications is going to give you the most direct and most sustainable path towards revenue, and then doing that thing, And now I will say that one advantage of the platform technology early on, and especially right now, because we're in the midst of this sort of cryobiology renaissance, is that the diversity does give you access to a huge array of interesting non-dilutive funding, right? So, maybe the company in the cryobiology space that has demonstrated this to the greatest extent would be one that Mehmet co-founded, Sylvatica Biotech. They've raised something like $20 million in SBIR grants through offering, commercial strategies on various different laterals that stem from the same platform technologies, right? So it's like you have two pots of money that you can access. Well, you have three pots of money you can access. There's non-dilutive funding, dilutive funding, and then revenue, right? So when you're in this pre-revenue stage, the breadth can be useful for non-dilutive funding, but is bad for dilutive funding. And then as you laser in on the application that is going to take you towards revenue, then you can make much more strategic decisions about dilutive funding for this particular application doing this thing and sublicensing out this one field of use of the patents, so on and so forth. So it's sort of this multi-tiered process. And I think we're sort of blessed right now to have access to a lot of interesting non-dilutive funding because we're at the sort of scientific forefront of a field that is going through a veritable transformation,
Great. That's a really good way to think about it, and this idea of the mechanistic parts of the funding and breaking it down into, again, when you're doing the science, the SBIRs are a great mechanism. There's obviously others as well that certainly we can help point people to as well. And then again, the whole ventures side is another totally different avenue before you get to revenue. A theme that you brought up, and I think it was brought up by all three of you, is this idea of that cryobiology is a very exciting space. Okay? And as an outsider, the question is, why should I, as an investor, for example, why should I be excited about cryobiology right now? Why is it in the growth phase? Why is it in the renaissance? We'll finish off with this question and let each three of you answer that question. I'll start with Kevin, go to Matt, and then we'll have Mehmet finish it up. But Kevin, you want to give your spiel about why we should be excited about cryobiology?
Ooh, there's a lot of things to be excited about.
Yeah.
I think, I mean,
From an investor perspective, right? Say, I'm investor looking at, P, maybe not PE, but a VC looking at, opening up something, a new space. I've heard about it. Sell me on the idea in about a minute.
yeah, I mean, from an investor side, I mean, it's just the growth of these potential new, the applications in cell and gene therapy and are just amazing, and really just the growth and the success of cell and gene therapies depends on cryopreservation to make it more widespread. Like like Dr. Toner said, we got to get away from this $500,000 per treatment option. And the only way to do that is to have allogeneic cryopreserved product that can be distributed. So it's extremely exciting, and there's a lot of potential money to be made in that area. And it's really also it's transformative. And that's just one area. I mean, the organ cryopreservation, if you would have asked people years ago if a full organ could be cryopreserved, the answer would have been no. And the fact that people like Bischoff are doing this, and all the exciting work in that area, I mean, and just the applications are endless, and in terms of the preservation of biodiversity and all these areas of transforming the way we think about food. It's just that there's so many things that are exciting about it, and also just the new technologies like are just also exciting, and people looking at nature and using natural molecules, and looking really into the nuances of what the mechanisms are of what's happening during freezing. We could go on and on, but it's exciting from a scientific and also from an entrepreneurial perspective. So that's my take. And I'm also not a cryobiologist, so it's all exciting for me. I just came on board and started learning, and it's more exciting than I would have thought from my previous experience.
Great. Thanks, Kevin. Man, I'll turn it over to you for your elevator speech.
Yeah, I'd go back to this dichotomy of the problem and the solution. We're in this really strange and I think infrequent, rare sort of set of circumstances where there's this huge problem that went unrealized for a long time, unrealized by the venture community and a lot of the medical community and whatnot, which is the problem being, let's call it just the challenge of cryopreservation of complex organic materials, right? And it just so happens that and of course it doesn't just so happen, there's a lot of people who made this happen, one of which is Mehmet and many others in our ATP-Bio community. But it just so happens that the world is starting to realize the magnitude of the problem at the same time that the scientific solutions are becoming available. And that never happens. Usually it's okay, we realize the problem, we put billions of dollars into the research, and by the time the solution emerges, there's hundreds or thousands of companies that have been working on it. There's a really competitive space, barrier to entry is high. Right now, we're in this magical wonderland where the magnitude of the problem has only been realized at a broad public level recently enough that there's a lot of low-hanging fruit that is achievable with recent advancements in the scientific, standard of the technology. So I think this conflation of marriage of circumstances makes for a really exciting domain. And I'll tell you, as someone who's talking to venture capitalists 10 times a week, every week for the past 3 years, even in the last handful of years, the enthusiasm in the venture community has just skyrocketed. I mean, 3 years ago when I was talking about, isochloric supercooling of a whole heart, I was talking to big med tech firms who were like, organ preservation, what? And now I'm getting cold calls saying, hey, our firm is looking to add something to our portfolio in organ preservation, right? And like Kevin said, this is one tiny example of the broader need, organ preservation, cell therapies, biodiversity stuff, food stuff, you name it. So it's the world is the venture world is starting to catch up here. And yeah, it's pretty exciting all around.
Great. Thanks, Matt. Maybe I'll leave the last word to you. You can maybe take this as practice for the NSF review committee coming through about why we should be excited about cryobiology.
Yeah, I mean, for the reasons Kevin and Matt said, basically, it's a very opportune time. The market has been developing for decades, but now it's maturing and the need is huge. I work as a consultant to every single tissue engineering company. I told every one of them, if they do not think about preservation of their product from the get-go, they're not going to survive. and they all went under. And enough of that happened, and the now market realizes that this is a real problem, and so they need to solve if they want to really put these products into marketplace. The other one is the science of cryobiology is much more mature. We know much more about it, so we can design things up, based on fundamental principles. It was a very empirical field in the past. And there are lots of new technologies, BioChoric, and so on and so forth. So they are really fueling these very diverse set of possible applications with these technologies. We can do a whole organ, and now we can also do one egg. And we have new technologies to do both of those. So I think the convergence of those three factors, unmet need and realization of this unmet, the importance of cryobiology in the venture capital and small companies, as well as pharmaceutical companies, science of cryobiology and the technology toolbox. Those are all now matured enough to have an impact.
Great. Thanks, Mehmet. With that, we are at time, but I just want to thank my speakers, Mehmet, Matt, and Kevin, for a really interesting panel today and discussion about their experiences. Hopefully it was useful to you as well in the audience, especially those young folks who have that entrepreneurial gene and want to exercise that gene and create something of their own. So with that, we'll go ahead and wrap it up. Again, stay tuned for more webinars from the ERC here. We'll have a series of both public and closed sessions coming up, but it's been hopefully as exciting for you as it was for you today. So thank you.
Thank